Hi, my name is Alyssa, and I'm a graduate student at Purdue on the St. Mary's Project. Our soils team looks at a variety of soil health indicators, but they all fall into three categories: chemical, physical, and biological. I'll talk about those today. I'll give an overview of the whole soils project. We'll talk about the data we've collected thus far and what it means for soil health. Our team takes samples from approximately 25 sites in the Nicholson and Weber Ditch subwatersheds, which are both a part of the Western Lake Erie Basin. Each site consists of 3 replicates. Each season, we sample from a different place in the field and use GPS coordinates to check the soil type against the soil survey. When soils are collected from the field, we first measure 3 10 by 10 meter plots, each 15 meters apart. Once the replicates are in place, the team will take 6 soil cores from each replicate. This core is 4 inches in diameter, and we sample to a 10 centimeter depth. We'll take 2 soil moisture tins, and finally, a bulk density from each replicate. And here we see Becca taking a bulk density sample, and this can be a really difficult process. Here, she's just finished hammering that piece into the ground, and she'll remove the sleeve, and she's going to cut between those 2 cylinders. The sample is that bottom cylinder, and if she cuts too much off, or if she's hit a rock or a root or the sample crumbles, she'll have to start again. Bulk density tells us a lot about the soil, its compaction, and the soil pore space that allows for movement of water, nutrients, and microbial habitat. When finished, tools are sterilized to ensure we do not cross-contaminate the soil samples between sites or take pests from one site to another. Samples are then taken into the lab where they are processed and stored. As I mentioned earlier, our lab measures a number of different soil health indicators. Biological indicators would include microbial respiration and enzyme activity, whereas chemical indicators would be things like pH, nutrients, and organic matter. Lastly, physical indicators would include textures and aggregate stability. In addition to these soil health measures, we've also collected farm practice data. If you're a participant in the St. Mary's Project and we take soil samples from your field, you've probably met Marianne or myself as we've surveyed farmers about cover crop and tillage data for the last 4 years. Using this information, we've been able to put participants into one of 3 categories. P1, or Practice Group 1, are participants with minimal use of conservation practice. That's a conventional tillage, and they do not grow any cover crops. P3 are farmers who use no-till and cover crops, as well as a nutrient management plan. P2 is a middle group of participants, maybe moving into conservation, or those who implement some conservation practices and not others. Using this practice data, we've been able to compare the data of the 3 groups. So what story does the data tell? The data collected thus far indicates a strong correlation between farmers of high conservation practice and microbial activity in their soil. This is substrate-induced response, and it measures the microbial reaction to glucose, a food source. The higher the microbial activity, the higher the nutrient turnover may be in the field, which leads to a biologically healthy soil. This is important to soil health because an increased active microbial population not only increases nutrient turnover, but also contributes to other soil health factors like soil aggregation, seen here, which also increased in the high conservation groups. This is aggregate stability as mean weight diameter, which is just the size of the aggregate. A strong aggregate stability would decrease erosion and nutrient loss, aid in better soil structure, and again, promote that microbial activity. Lastly, phosphorus sorption increased with high conservation practice. This is a soil's ability to retain phosphorus, and it means limiting movement of phosphorus off-site. This is a particularly interesting find and one we're looking into further. Thanks for watching. If you have any questions about soil health or the St. Mary's Project, I can be reached at abesser@purdue.edu.